Hello, this is Dr. Aruna Saman, and today we are going to discuss in this video protein synthesis. Proteins are made up of amino acids. They are the structural components, enzymes, and hormones. The living cells need to synthesize new protein molecules, and all types of proteins are synthesized by the cell intracellularly. The process of protein synthesis includes two major steps, which are transcription and translation. The central dogma of molecular biology was postulated by Crick in 1958. It explains the molecular path of flow of genetic information. The unidirectional flow of information from DNA to RNA by the process called transcription and RNA to proteins by the process of translation is referred to as central dogma of molecular biology. In this dogma, it is explicitly said that information goes only in one direction and never in reverse direction. But the present concept of central dogma given by Temin and Baltimore in 1970 suggests that it is possible to transfer the information in reverse direction also. That is, from RNA to DNA, the process is reverse transcription, and then of course from DNA to RNA by transcription and RNA to protein, the translation process. This is observed in retroviruses or riboviruses such as viruses causing AIDS. The two major steps of protein synthesis as mentioned earlier, transcription and translation. The transcription occurs in the nucleus and translation in the cytoplasm on the ribosome. In the transcription from DNA, the information is transferred to RNA, and RNA formed during this process is called primary transcript, which undergoes the maturation to form to become messenger RNA. So this is transcription, and then the messenger RNA form comes out of the nucleus through nuclear pore. It gets attached to the ribosome. On this messenger RNA, as per the codons present, the amino acids are brought by TransferRNA, and these amino acids get connected by peptide bonds to form protein. So thus transcription is from DNA to messenger RNA in the nucleus, and translation is from messenger RNA to proteins on the ribosome. Let us understand the process of transcription in detail. The process of transcription is copy genetic information from DNA to RNA. So information is taken from one of the strands of DNA. That strand is called template strand. And the entire process of copying this information is called transcription. This transcription process occurs in the G1 and G2 phases of cell cycle. And once the transcription takes place, the messenger RNA gets formed. Then this information goes to the ribosome in the cytoplasm. Here we can see the DNA with transcription units. The transcribed segment of the DNA shows a region called promoter. Then there is structural gene, and then there is a region called terminator. These together form transcription unit. This transcription unit has promoter, which is located at 5' end of the coding strand or sense strand. It is a DNA sequence that provides a binding site for RNA polymerase. RNA polymerase gets connected at the specific promoter. And then what is structural gene? The structural gene is a segment that has information about the protein synthesis. This information is in the triplets of nitrogen bases, which are called codes. The DNA strand having 3' to 5' end polarity contains the code and it acts as a template strand. The RNA polymerase catalyzes polymerization in 5' to 3' direction. The other strand of DNA having 5' to 3' polarity is complementary to the template strand. It is called sense or coding strand. The sequences of nitrogen bases in the messenger RNA will be almost same as in the strand, the coding strand, except the messenger RNA will have the thiamine replaced by uracil, whereas the terminator is located at 3' end of the coding strand, that is downstream. It marks the end of the transcription process. We will understand the transcription process now in detail, the mechanism of transcription. The 3 steps involved in transcription are initiation, elongation, and termination. The enzyme RNA polymerase binds to the promoter at the template strand towards 3' end. The RNA polymerase then moves along the DNA and that causes the local unwinding of the DNA into 2 separate strands in the region of structural gene. So this way RNA polymerase moves along the template strand and the 2 strands they separate. And then the next step occurs, that is elongation. When the 2 strands separate, naturally the nitrogen bases of the template strand, they get exposed to the nucleoplasm. The exposed nitrogen bases attract complementary ribonucleotides from the nucleoplasm, and a single stranded RNA will get formed against this exposed template of DNA, that is structural gene. Let us understand it here. These are the 2 strands separated with the help of RNA polymerase. This is the template strand with exposed nitrogen bases. We can see the actual nitrogen bases here. Then the RNA polymerase got attached. It will move along this template strand, and when it will move, it will attract the complementary ribonucleotides which will form messenger RNA. So here we can see the RNA polymerase moving along and the messenger RNA is getting formed here. This messenger RNA, of course, is not matured messenger RNA. It is called primary transcript. The next step occurs, and that is termination. During termination, here we can see the primary transcript which is formed against the structural gene got dissociated from that structural gene. Once it gets dissociated, then again the winding of the DNA molecule will take place. It will get spirally coiled and will become double helix. The primary transcript or heterogeneous nuclear RNA is not a functional RNA because it has, we can see here, both exons and introns, and therefore this need to undergo the process of maturation, and this maturation process involves 3 steps: capping, splicing, and tailing. In capping, the primary transcript gets methylated guanosine triphosphate at its 5' end, and then in splicing what happens is the introns of the primary transcript are removed and only exons are kept. and these exons get connected with the help of DNA ligase enzyme. So these introns get removed and only exons are left behind and they get connected. And thus the primary transcript slowly gets converted into messenger RNA with the last step of tailing, and that is polyadenylation takes place at 3' end. And thus the tailing also gets over and now it is fully processed heterogeneous nuclear RNA called messenger RNA. Once the messenger RNA is formed, it moves out of the nucleus and goes into the cytoplasm, and the next process of protein synthesis, that is translation, occurs. Translation is the mechanism in which codons of messenger RNA are translated and specific amino acids in a sequential manner from the cytoplasm are brought on the ribosome and they are connected on the ribosome by peptide bonds. The different things necessary for this translation are amino acids, messenger RNA, transfer RNA, ribosome, ATP, magnesium ions, enzymes, the elongation, translocation and release factors. Let us understand few of these things in detail, like amino acids. They form the raw material for protein synthesis. About 20 different types of amino acids are available to form proteins. They are there in the cytoplasm. DNA in its structural gene contains information which exact amino acids to be assembled on the ribosome. So here we can see the DNA having this information. This is called DNA code and it is in the form of triplets of nitrogen bases. This information is there in the nucleus in the DNA and this is taken from DNA by single-stranded RNA called messenger RNA. So here we can see the messenger RNA. It has taken down the information using its own ribonucleotides and there the triplets of nitrogen bases are called codons. These triplets, they are representing different amino acids. And these amino acids get connected by peptide bonds to form polypeptide molecule or protein. The messenger RNA, it is a straight and simple RNA formed from DNA by the process called transcription. We have already discussed the transcription process. It carries the information from DNA to ribosome about the sequence of amino acids to be assembled. It has 5' end and 3' end. A sequence at 5' end is called start codon. It is AUG and it always specifies the amino acid methionine. The codon at 3' end is stop codon or termination codon. It is either UAA, UAG or UGA. They do not specify any amino acid and hence also called nonsense codon. The transfer RNA, it is also non-genetic, single-stranded but peculiarly folded RNA. At its one loop, it has triplets of unpaired bases called anticodon. If you remember, the triplets of nitrogen bases in the DNA are called codes. In the messenger RNA, they are called codons. And now in the transfer RNA, the triplet of nitrogen bases present at this loop is called anticodon. At one of the unequal ends, at the 5' end, at the free end, 5' end, it has guanosine nucleotide and at 3' end, there is a sequence of CCA nucleotides. These are the fixed nucleotides found on the transfer RNA. The anticodon of transfer RNA is always complementary to the codons on messenger RNA. The transfer RNA carries specific type of amino acid always at the CCA end to the ribosome. Thus, it translates the coded message of messenger RNA in terms of amino acid. That is the function of transfer RNA. So here we can see the messenger RNA gets attached to the ribosome and it has the message in the form of triplets of nitrogen bases. These are the nitrogen bases, and complementary to these triplets, these codons, the anticodon bearing transfer RNAs, they keep on bringing the amino acids, and these amino acids get connected by peptide bonds to form proteins on the ribosome. Ribosome, they serve as site or work table for protein synthesis. Each ribosome contains larger and smaller subunits. These subunits occur initially separate in the cytoplasm. Only during protein synthesis, these two units, they get associated together with the help of magnesium ions. A ribosome has one binding site for messenger RNA and 3 binding sites for tRNA or transfer RNA. These 3 binding sites are P site, which is also called peptidyl tRNA site. Then there is A site, also called amino acyl tRNA site, and there is E site or exit site. Only the first tRNA amino acyl complex directly enters the P site of the ribosome. Subsequent tRNA amino acid complex always enters the A site of the ribosome. When the ribosome moves on the messenger RNA, we find that the position of the transfer RNA shift from the P site to E site and A site to P site. Let us see here. Now this is the ribosome which is shifted on the messenger RNA. And see the first transfer RNA which brought methionine. It was at the P site, it got shifted to E site. The second tRNA, it also brought the amino acid and it entered at A site. But when ribosome shifted on the messenger RNA from A site, the second transfer RNA got shifted to P site. Now the A site has become vacant. So the third transfer RNA will come to the A site. By that time, the first transfer RNA will be released and again the ribosome will get shifted. So this is the function of ribosome. The mechanism of translation in detail, it involves 3 steps: initiation, elongation and termination. The initiation step, it involves activation of amino acid using ATP. The amino acids which are there in the cytoplasm, they are first activated by ATP. Then small subunit of ribosome binds to the messenger RNA at 5' end. The initiator codon AUG, with this the initiation process starts. This AUG always attracts the messenger RNA having the complementary nitrogen base, and that is UAC. This transfer RNA always brings methionine amino acid. The larger subunit of ribosome then joins with the smaller subunit after the attachment of the first transfer RNA which has brought methionine. After that, the larger subunit of ribosome joins this smaller subunit with the help of magnesium ions. And the transfer RNA occupies P site. This is the only time where the transfer RNA right in the beginning is at the P site. The subsequent transfer RNA will always enter at A site. But this is the first transfer RNA and the only transfer RNA which gets attached at the P site. The next transfer after initiation, there will be elongation. And in the elongation, we will see the first transfer RNA is already there with its amino acid. Now the second transfer RNA will come at the A site with its own amino acid. The next step is elongation. The activated amino acids, they bind with the transfer RNA to form transfer RNA amino acyl complex, and that complex comes to the ribosome, and that way activated amino acids are added one by one to the first already which is there on the ribosome, the methionine. To this amino acid, the further brought amino acids get connected. It involves three-step cycle. These three steps will occur in a cyclic manner. Step 1. Now, as said earlier, at the A site, P site, already the transfer RNA was there in the initiation step. Now this is elongation step. The next transfer RNA has come and it has brought another amino acid. Amino acyl tRNA molecule has entered the ribosome at A site, because at this A site there is present a codon, and complementary anticodon bearing transfer RNA has brought the specific amino acid. The anticodon and codon, anticodon of transfer RNA and codon of messenger RNA, they bind with each other by hydrogen bonds. In the step number 2, the amino acid on the first initiator tRNA at P site and amino acid at tRNA at A site, they join. Already come methionine and now brought next amino acid. They are joined with the help of peptide bond, and the active enzyme is ribosome. And in the step number 3, which is very important step, and that is translocation, the ribosome, it moves along the messenger RNA. And when ribosome moves, the position of the transfer RNA gets shifted. Now you can see in this diagram, the methionine which was present at the P site, it has come to the E site. And from E site, that transfer RNA will be released from there. But already what methionine it had brought got connected with the second amino acid, and as a result the A site has become vacant. So both subunits of ribosome, when move along the messenger RNA, we can see the position of the transfer RNA getting shifted. From E site, the transfer RNA, first transfer RNA gets discharged. The second transfer RNA was there at the A site, now it comes to the P site. And at A site now, the third transfer RNA comes with another amino acid. And once again, the ribosome will shift and the first step will be repeated. So as we said earlier, that the three steps occur in a cyclic manner. So once the ribosome shifts, then again you can see the amino acyl tRNA molecule enters the ribosome at A site, and then the hydrogen bond formation takes place. The whole process will get repeated. And then the last step, and that is termination. In the termination step, one of the stop codons, maybe UAA, UAG or UGA, is exposed at the A site, and then it does not read or is not joined by any anticodon of transfer RNA. It does not mean any amino acid, and therefore now the release factor binds to this stop codon. So in the next step, we will find that release factor got connected to the stop codon and thereby the terminating translation process. And by that time, polypeptide is now released, is formed. It will be released in the cytoplasm. And in the last step, the two units of the ribosome, they separate, and the releasing factor or the transfer RNA, messenger RNA, they are all released in the cytoplasm. But the most important thing is the chain of amino acids with the help of peptide bond. So polypeptide chain or protein got formed. So this is the way entire process of protein synthesis takes place. If we study this process in a gist, then we realize that the entire process occurs in two steps, transcription and translation. The transcription in the nucleus, translation in the cytoplasm on the ribosome. In the transcription from DNA, single-stranded RNA is formed. The three steps are involved in that: initiation, elongation and termination. In the termination, where maturation of the primary transcript takes place and it becomes messenger RNA, that processing or maturation occurs in three steps: capping, splicing and tailing. And once the messenger RNA is formed, then the translation. Again, three different steps are understood here: initiation, elongation and termination. And here, especially in the elongation, the three steps keep on occurring in a cyclic manner. With every cycle, an amino acid get connected with already present amino acids, and finally with the stop codon, the termination will take place. So this is the way finally we get the protein formed in the cell. We have come to the end of protein synthesis discussion. Thank you.